Hello, and welcome to the Asthma and Allergy Foundation of America's latest education program titled Mastocytosis and Other Mast Cell Disorders. This program is developed independently by AAFA and made possible by Blueprint Medicines. We are so glad you are here today. My name is Stacy, and I am the Public Health Manager here at AAFA. And today, we have a great program for you, but before we dive in, I want to tell you a little bit more about the Asthma and Allergy Foundation. We aim to be a trusted ally of the asthma and allergy communities, and we are dedicated to saving lives and reducing the burden of these diseases through support, advocacy, education, and research. And without you, our community, we wouldn't be here. So thank you for everything you do for those everywhere with asthma and allergies. Again, my name is Stacy, and I am so happy to be here and happy to be your host for the program. We are also going to be joined by Dr. Matthew Greenhawt. Hi, Matt. How are you doing today?
Good. Thanks, Stacy. How are you?
I'm doing great. Thank you. Dr. Greenhawt is our Chief Medical Officer here at AAFA, and he will moderate the session today with Dr. Lawrence Schwartz. So let's go through our agenda really quickly. We're going to be discussing mast cells and mast cell diseases, mastocytosis and symptoms, and why they can be confusing to diagnose, along with what kinds of tests your provider might ask to accurately diagnose the condition. We will discuss available treatment options and how to work with your provider to develop a treatment plan. And finally, we will discuss support and resources that are available. But first, Dr. Larry Schwartz is our speaker today. Not only does he sit on our MedSci board here at AAFA, but he is a distinguished professor of medicine at Virginia Commonwealth University. He is a translational research investigator who has made fundamental discoveries about human mast cells. These include identifying different types of mast cells and tryptases. He has also produced therapeutic and clinical tools that facilitate the diagnosis of systemic mastocytosis that have been FDA and World Health Organization approved, as well as anaphylaxis, the discovery and diagnosis of mast cell activation syndromes, and of hereditary alpha-tryptosemia. Dr. Schwartz, you'll probably have to go over that word for me in the future, and the monitoring of mast cell cytoreductive therapies. By caring for patients with such disorders and being involved in related basic translational and clinical research, he has a unique perspective to share. We are surely honored to have you here, Dr. Schwartz, and we thank you for your expertise. We are so proud that you are here. Here are our disclosures. And now I'm going to hand over the program to Dr. Greenhawt and Dr. Schwartz. Matt, please take it away.
All right. Thanks, Stacy. So, Dr. Schwartz, you are as a trained allergist. I've been following your work for years. It was instrumental in my learning as a fellow, and then in my application as an attending physician. I used to be really associated with the mast cell program at University of Michigan for a number of years. So this is a great honor, and you and I have known each other for a while. You are one of the biggest names in mast cell disorders in the world. You've authored numerous papers. You've given many talks all over the world. You really helped define this field by identifying not only the types of mast cells, but discovering sort of the types of human tryptase that we have, which is our main biomarker for looking at mast cell disorders. You've helped characterize their activities, and you've helped, really develop the immunoassay field, which every time, for the patients out there, every time you go to the doctor and you have one of these tests run, this is Dr. Schwartz's work, and this is his direct contribution to the field. So what is it that really ignited your passion in mast cell disorders and sort of drove you to help develop all these wonderful tools that we have?
So in 1978, with a background in biochemistry and training in internal medicine, I went to Boston to do a fellowship in rheumatology and allergy immunology. And as I was introduced at that time to mast cells in laboratory, I realized that allergic reactions were diagnosed solely by symptoms, and that struck me as being somewhat imprecise. At the same time, our colleagues in cardiology and hepatology were using enzymes measured in the blood as biomarkers of what was going on in the tissue in those organs. So I wondered if mast cells, when activated, might also release enzymes that could be measured in the blood that would give us insight into their involvement in different diseases for which they were involved. So in 1981, my laboratory discovered and named an enzyme that came primarily from mast cells named tryptase, and then developed antibodies against it that were eventually used to develop the assay that is used today. And so we've learned using that assay that serum tryptase levels can use as a biomarker to help, not by themselves, but to help diagnose mast cell activation disorders such as anaphylaxis and mast cell activation syndrome, and also help with the diagnosis of systemic mastocytosis, hereditary alpha tryptosemia, and also the therapeutic reduction of mast cell burden in some of those diseases as well.
Yeah, I mean, this is great. I mean, you take, in allergy, we don't necessarily have a lot of biomarkers. If you're having chest pain, you go to the hospital, you have something like a troponin level that gets checked, or like you mentioned with liver disease, there are certain enzymes there. This is our own marker that really does help us understand sort of when a patient has symptoms to make a better fit. So, all right, so let's start at the beginning. And can you give us your perspective on sort of what the mast cell is?
So if you think about the bone marrow, that's where there are progenitor cells that can give rise to a variety of blood cells, one of which is the mast cell. And then mast cells that are derived from those progenitors, they can either complete their maturation to mast cells, become mast cells in the bone marrow, and then they'll stay there, or those progenitors can come out of the bone marrow and settle at different tissues throughout the body and complete their maturation there and become mast cells. Those mature mast cells are characterized by expressing receptors on the surface of those cells, and I think you can see in green the IgE molecules, which bind very tightly to those receptors on mast cells. There's also another protein on the surface of mast cells called KIT that is very involved in the development of mast cell, its survival after it develops, and also facilitates but doesn't cause the activation of mast cells. And then you see those kind of large circle in the oval type structure in the middle of that cell, and that's just the nucleus, which you see in all cells. But then outside of the nucleus, you see those blue spheres. Those are the secretory granules of mast cells, in which some of the mediators are stored, one of which is histamine, which I think everyone's heard of because we take antihistamines to block its effect, and another one is tryptase. So that characterizes this type of cell, and having that IgE on the surface of the cells means that IgE can bind to an allergen that it recognizes as an antibody, and that allergen, if it aggregates that IgE on the surface of the cells, will activate those cells. So if we see the next slide, we'll see that once a mast cell is activated, and let's just imagine that allergen has bound at least two of those IgE antibodies, so it's aggregated it on the surface of mast cells, that sends a signal inside the cell to begin to produce and secrete mediators. And mediators are chemicals or substances that are the signs and symptoms associated with mast cells when they're activated. And so some of them, like histamine and tryptase, are stored inside those granules and come out when the granules are released or the cell degranulates. Other mediators, like at the bottom of the screen, the prostaglandin D2 and leukotriene C4, are lipid chemicals that are not present in the resting mast cell, but when the mast cells are activated, they're newly generated and secreted by those cells. And so these mediators and others will cause the symptoms that are associated with mast cell activation, such as flushing, swelling of the itching, shortness of breath, dizziness, and fainting associated with low blood pressure. So in this slide, it emphasizes that mast cells can develop and reside in many parts of our bodies, as shown here. And the importance of that is these naturally occurring mast cells are waiting to be activated. And you can imagine the symptoms might be quite variable if mast cells only in the eyes are activated versus in the nose, versus in the airways, or versus in the gut or the skin. And the symptoms can very much vary depending on where mast cells are getting activated. So how are mast cells activated? So we've talked a little bit about this, that in allergic reactions, it's an allergen that binds to IgE that's already bound to the mast cell and aggregates that IgE on the surface. And that act causes a signal to go inside the cell. It causes it to start secreting mediators. But allergy is not the only way mast cells are activated, and not only through the IgE process. And so there are a variety of ways that mast cells can respond to environmental stimuli. Even physical stimuli in some patients can activate mast cells, whether it's scratching or rubbing or vibrating the skin or challenging it with heat or challenging it with cold. There are a number of physical stimuli that in some patients can activate mast cells too.
Yeah. I think that's an important point, because sort of the end result to the patient is, I started wheezing, I started itching, I have hives, right? And when they come to see us as patients, it's up to us to sort of dissect that a little bit further. Well, was it directly triggered? Was this from, did you eat something you were allergic to? Did you inhale something? Or, was it a direct effect? And while we have different names for that, to the patient, they feel the same thing. And for us, technically, it makes a big difference in sort of how we can test and assess, because these direct things, a lot of patients sometimes leave a little dissatisfied with us when we can't necessarily test to certain things like that, antibiotics or whatnot, that are notorious for causing these sort of more direct reactions or whatnot, versus something that was allergen triggered. So I think that was a really good explanation that you gave of sort of just the different ways that these cells can sort of be sort of motivated to fire off and cause the symptoms.
So this slide lists a portion of the many symptoms that might be included in an allergic reaction, affecting itching, sneezing, runny nose, a variety of rashes, particularly hives. Could be stomach cramps, nausea, vomiting, diarrhea, pain in some areas, swelling of the tongue, coughing or throat closing from swelling in the airways, wheezing from the muscle contracting around the airways, feeling faint or lightheaded, or even blacking out from a drop in blood pressure. And some patients in the more severe reactions have this sense of impending doom at the beginning of a severe systemic allergic reaction.
Yeah, and just fielding complaints from patients, or watching reactions that I myself have provoked in trying to diagnose or whatnot. Yeah, I mean, the amount of different symptoms that somebody can feel, it's impressive, right? And it runs the gamut of pretty much every organ system. And our job is trying to figure out, was this triggered by an allergen? Was this triggered through another process? Or, sometimes these are not necessarily, like a runny nose can be from a number of things, like feeling faint or whatnot. Some of these cross over into non-allergic, non-mast cell symptoms. So, it becomes hard for us to figure out. It's easy to pop some of these symptoms into Google or ChatGPT or whatnot, but the differential, what we call the differential diagnosis of sort of, what's the most probable source of this? And, we develop a list from highly likely to least likely and stuff. And it's, these are all the things that, in an office visit, we're listening to you and we're running all these sort of processes in the background, trying to figure out, how do we think this happened? What's the source or whatnot? So if you've ever seen us sort of looking dumbfounded in the visit when you're explaining sort of your history of all these symptoms, that may be why, because I mean, it overlaps with a number of different things, and it's just sort of a very impressive response.
Sure. And not shown on this slide are important questions relating to the timing of these events. How long did they occur? What time of the year they occurred? What might have triggered them, and so on. And that all gets massaged into what we think is the most likely diagnosis. So when one has systemic allergic reactions. They're often referred to as anaphylaxis. And we'll talk a little bit about how anaphylaxis is defined, but it's a sudden onset, rapidly progressing. It can be severe and it can be potentially life-threatening allergic reaction that comes on quickly. In rare cases can cause death. And importantly, to recognize that not all allergic reactions are anaphylaxis, particularly if they just occur in one organ system. Symptoms of anaphylaxis usually involve more than one organ system, and so might include the skin and the GI tract, or the cardiovascular system and the skin, for example. And over here on the right, you see that 90% of anaphylactic reactions in infants and toddlers include involvement of the skin, almost 60% swelling in a variety of areas of the body. About half have GI symptoms. Just under half have coughing and wheezing, so respiratory symptoms. And sometimes they have symptoms just around, including around the eye, recognized as rubbing from itching and redness, maybe from the rubbing. That's hard to know sometimes.
Yeah. And there are other things too that, AAFA has done a lot of work on this in the last couple of years. And the field has recognized that sometimes the infants and toddlers, the extremes of ages, maybe have slightly different presentations than somebody sort of in the middle of life or older child. With the infants, behavioral changes, becoming quiet, becoming clingy, sometimes pulling. Refer you to the website to go over some of the signs and symptoms, but it's important recognition. In a shameless plug, AAFA has just released an updated anaphylaxis plan that has very specific signs and symptoms tailored towards the younger children. So I invite you to check that out too. But it just underscores that sometimes it's a common symptom, but it may have slightly different versions, and depending on who you are and what happened. And that's actually a nice segue into this, because this slide, you're going to go over sort of why we have different criteria here.
Sure. And so a number of organizations have developed clinical criteria for trying to get either a more precise or a more rapid diagnosis of anaphylaxis. And I would say that the Brighton criteria are trying to use specificity, not necessarily timeliness, to try to diagnose anaphylaxis. The National Institute of the Allergy and Infectious Diseases was trying to develop criteria that could be used rapidly, say in the emergency department, so that treatment could be instituted based on a rapid diagnosis. And then the World Allergy Organization tried to bridge these two together to try to get some of the best of each. But you see that all of them require a sudden onset and rapid progression of signs and symptoms for anaphylaxis, and involving, in most cases, with few exceptions, multiple organ system involvement. So the Brighton criteria required skin, and then as a second criteria, you could have symptoms related to the cardiovascular system, airways, or gut. The NIAID, you could have either skin or heart or airways or gut. And then the WOW required skin and either heart or airways or gut. So they went more with the Brighton criteria. But both NIAID and WOW made exceptions to the multi-organ system involvement. And that is, if somebody was exposed to a known allergen and the symptoms followed soon after that, then the NIAID said, just with low blood pressure or cardiovascular symptoms alone, you could make a diagnosis. And WOW said, well, we agree with the low blood pressure, but also airway alone could be used. So you see a little bit of variability, and you can also imagine that some of these clinical criteria are somewhat imprecise. Using these clinical criteria, there have been a non-biased estimate of, in adults, the lifetime prevalence of anaphylaxis is somewhere between 2 and 8%, depending on the stringency of the criteria that are used. But there is a differential diagnosis that one might consider when people present with many of these symptoms. And I think we'll talk about that in the next slide.
Yeah, I mean, I think that's a great description of sort of the evolution of these different criteria and whatnot. The hardest thing to explain to somebody is that depending on where you are in the world, the same symptoms could be anaphylaxis based on one symptom, one criteria, and not anaphylaxis based on the other. And depending on, like, Brighton was really, and with disclosure, I'm part of the working group that has helped redevelop the criteria for that. These are meant to be applied in areas where there's not necessarily somebody with, allergy training. It's meant for somebody who, with lower training. Think, in parts of the world where there's not as much access to specialists or other services, or in parts of the world where people, even without medical training, are giving vaccines or things like that. It's supposed to give you sort of a ranking order, like how probable do I think this is, whereas the other criteria really developed to not miss things, sort of capture it as, yeah, I think that this may fit it, and kind of figure it out later and stuff like that. Not to complicate it, but there's actually a fourth set of criteria coming out from the Galen organization or whatnot, and sort of there are lots of friendly and sometimes not so friendly academic debates over which criteria should reign supreme or something like that. But we're all of these are trying to get to the same thing. They're trying to describe it in the best way to tell, with high probability if you had anaphylaxis or whatnot.
Great. So given some of the inherent imprecision in using clinical criteria alone, this gives us a differential diagnosis or other disorders unrelated to mast cell activation that could produce overlapping or similar symptoms. One are a variety of stress-related responses, vagal reactions, panic attacks, vocal cord dysfunction, autonomic dysfunction with postural orthostatic tachycardia syndrome or benign flushing disorders, not so benign flushing disorders due to carcinoid syndrome or neuroendocrine tumors, angioedema due to activation of the kinin pathways in the body and resulting in swelling from hereditary angioedema or acquired angioedema defects in this pathways leading to bradykinin production, but not mast cell activation. I mentioned earlier that complement products can activate mast cells, but they can also produce some of the same signs and symptoms that mast cell activation causes without activating mast cells as well. Some foods, particularly fish that are stored perhaps improperly, the bacteria in those fish can make histamine, and then when they're eaten, you get an acute disorder from eating that fish, kind of a food poisoning due to the histamine that looks like it could be an allergic reaction, perhaps in some people. The name is called scombroidosis, but it doesn't involve mast cell activation. A variety of pulmonary or cardiogenic disorders may present with what looks like a mast cell activation event, or other shock syndromes due to septic shock or toxins that cause a drop in blood pressure, say, and one wonders whether that is due to mast cell activation. But these septic proteins or toxins don't activate mast cells, but they cause overlapping signs and symptoms. So this leads to a question is, can we be a little bit more precise if we include measurements of biomarkers with the appropriate signs and symptoms?